You are most welcome, our dear Senior 5 biology students. We are going to discuss membrane proteins and specifically look at the functions of membrane proteins as the last bit of the notes that we had taken in class. After this, that means our cytology will be totally complete and you can go ahead to start looking at other components of your biology course. Let us take a tour of the membrane together and look at component parts of the plasma membrane. In our picture, we have what they call the peripheral protein. These are proteins that are at the surface of the plasma membrane. They are not tightly bound to the phospholipids. They are loosely attached and they do not penetrate deep into the plasma membrane. Above this peripheral protein, we see a phospholipid. The phospholipid has a head and two tails. We shall mention something about the phospholipid. If we move on, we see two other types of proteins, the channel protein and the carrier protein. These two span the plasma membrane. They move from one side of the plasma membrane to the other side. They are referred to as integral proteins. If you compared these to the first one that I showed you, the first one is at the surface, therefore it is peripheral, and it is therefore called a peripheral protein, while the other two span across the plasma membrane. They are integral proteins. Now, closely look at them and notice that the two have certain differences in terms of their shape. They have a different shape. One seems to have a channel, the other one, the channel seems to be closed. That means these two proteins have specific functions that they perform to ensure that the plasma membrane performs its general role. Then when you look at this other type of protein, it does not penetrate deep into the plasma membrane, but it has a carbohydrate attached to it. Now, this type of protein is a conjugate protein. It is a protein with a non-protein part attached to it. It is referred to as a glycoprotein. It's also part of our plasma membrane. Then below this glycoprotein, we notice another molecule there. It is actually a steroid, and its specific name is cholesterol. Cholesterol has a role it plays or performs in the plasma membrane. It ensures that the plasma membrane is stable. Okay, then closely looking at the phospholipid, follow that arrow to get the detail there. This phospholipid has a phosphate head that is polar and therefore hydrophilic. A hydrophilic substance is one that is water loving. So this portion of the phospholipid is water loving. It is a part that can be in close contact with the aqueous medium. Okay, it could be tissue fluid, it could be blood, it could be anything that is aqueous. Okay, because that part is water loving. Then you have two tails. They are fatty acid tails. They are non-polar and therefore hydrophobic. Something that is hydrophobic is water hating. So that component, when inserted into our plasma membrane, should be away from the aqueous medium. So the arrangement in this model, we shall mention, you see that the hydrophobic tails should be away from the aqueous medium. They should be arranged in such a way that they are away from the aqueous medium. So today, our dear students, we are discussing the functions of the membrane proteins. But we need to know what the plasma membrane is. A plasma membrane is the edge of life. It is the boundary that separates the living cell from its non-living surroundings. Okay? Remember that the cell carries out a number of chemical reactions in it, and it will need raw materials. These raw materials, in the form of nutrients, come from the surrounding. So that means that these raw materials must cross the plasma membrane and find their way into the cell to support the physiological reactions that we shall learn about in the course of our biology study in Senior 5 and Senior 6. Now, this plasma membrane controls traffic into and out of the cell. Therefore, it has selective permeability, allowing some substances to cross it more easily than others. Now, charged particles do not easily cross the plasma membrane, but these charged particles might be required in the cell or inside a cell organelle. For example, when you break down glucose into pyruvate, pyruvate is a charged particle. Now, how does pyruvate enter inside the mitochondrion such that it can be involved in the Krebs reactions and then later on into the electron transport system? How does pyruvate go in? Yet it is charged and it cannot cross a membrane. So there must be ways in which some of these very important materials enter the cell. And that is why we are discussing membrane proteins today. The ability of the cell to discriminate in its chemical exchanges with the environment is fundamental to life. Okay, students, in the next slide, we have 3 guiding questions, but I would encourage you to use your question banks. In Senior 5, you may not have gotten yourself a question bank, especially UNEP, but please, I'm encouraging you during this time at home, get yourselves question banks and start looking at questions as you revise. Use the textbooks, use the materials we are sending you, and then look for questions related and see if you are able to answer some of these questions. Take note of some of the commands in this question such that you avoid misfiring. Now, for example, in the first question, outline the various functions of membrane proteins in the plasma membrane and other membranes of various organelles. The same question is repeated but differently. The question is now, explain the role of membrane proteins in the plasma membrane and other membranes of various organelles. The first one, outline. This one says explain. So an outline, that particular answer will not be so detailed. You might use bullets and you simply say this part does this part does that. Okay, but when they ask you to explain, you go a little bit deeper and provide more information that is presented logically and precisely. You avoid beating about the bush. Then the third question is for revision also. Explain why the plasma membrane is described as fluid mosaic. Okay, so closely look at this picture to see exactly where the plasma membrane is. We have a cell there, and then you have a boundary. That boundary is marked by the plasma membrane. membranous organelles like the nucleus are also surrounded by membranes. Okay, so the composition of those membranes with the plasma membrane is basically the same. They all have membrane proteins in them. What is the function of those membrane proteins? Looking closely at the cell membrane, we see the component parts. It looks interesting. Using the light microscope, we cannot see that detail. But using the electron microscope, we are able to see more detail. Why? Because the electron microscope uses a beam of electrons that have shorter wavelength than light. And once you have shorter wavelength, then the resolution is far better and allows you to get detail or to study detail, and we're able to see the component parts of the cell membrane that you're calling the plasma membrane. Now, if you go into more detail, just a portion of that plasma membrane, we notice that this plasma membrane is made up of a phospholipid bilayer. Okay, now the tails point inwards, then the heads that are hydrophilic point outwards. Okay, now if you look closely, you have the phospholipid there with the head which is hydrophilic, and then the tails which are hydrophobic or water hating. Now the arrangement of this plasma membrane was controversial for some time, but as the microscopy improved, different models were arrived at until we have the most recent and widely accepted model of the plasma membrane. Now, before the fluid mosaic model were other models by other scientists. For example, in 1895, Charles Overton postulated that membranes are made of lipids. Now, for this guy, he did not know that membranes have proteins as well. But remember, we must excuse him because the microscope was not well developed by then. So for him, through experiments, he concluded, no, this membrane is made up of only lipids. However, 20 years after his postulation, it was found that in the red blood cells, proteins were as well present. So that one overturned the other gentleman's model. Then in 1925, two Dutch scientists, Gotha and Grendel, reasoned that cell membranes are actually phospholipid bilayers, two molecules thick. But they did not account for the positioning of the proteins. So that model also quickly faded away. In 1935, Davson and Danieli then tried to place the proteins. For them, they made a serious mistake. We are saying serious because today, at least we have the, I don't know what you should say, the merit. We are a little bit advantaged because now we can use the electron microscope to study more detail. But these gentlemen, Davson and Danieli, thought that the proteins actually sandwich the phospholipids, and that model later on with more study had problems. Okay, you need to identify some of those problems, but the most acceptable today, that model that is most acceptable today, is that one for S.J. Singer and Garth Nicholson, and they proposed their model in 1972 to explain the structure of the plasma membrane. And according to them, biological membranes are essentially quasi-fluid structures in which lipids and proteins are organized in a mosaic manner. Okay, now according to this model, the fluid mosaic model, there is a layer of lipid molecules which are phospholipids in nature. I mean phospholipids in the center. The protein molecules surrounding the phospholipids are arranged in different ways in different regions of the plasma membrane. So you don't have a systematic way of arrangement. They are arranged in different ways within the plasma membrane. Okay, so that model, the fluid mosaic model, shows that proteins are dispersed and immersed in the phospholipid bilayer, which is in a fluid state. Okay, electron microscopy reveals that some of the proteins penetrate into the hydrophobic interior of the membrane, which is impossible with the Davson-Danieli model that I mentioned a little bit earlier. Some of the proteins, like I've said, penetrate right through the lipid layers. They traverse, they penetrate right through from one side to the other. They span the plasma membrane, and they are referred to as intrinsic proteins, intrinsic or the integral proteins. Now, some, like the other one you saw with the carbohydrate attached, partially penetrate on either side of the lipid layer, but they don't go through it. Then you have these other, which we call the peripheral proteins, that are on the surfaces. We are saying that some of these proteins lie outside the lipid layer on either side. They don't penetrate, they are loosely attached, and they are referred to as extrinsic proteins. Members, please take note of that as you revise. Okay, intrinsic proteins, they span across, then the extrinsic proteins are basically surface. Okay, but all of them have functions that are very vital for the functioning of the cell. The hydrophobic tails, remember, hydrophobic, water hating. So the hydrophobic tails of the phospholipids face each other internally, whereas the hydrophilic heads, those that are water loving, face towards the protein layers. Okay, now the protein layers we are talking about, especially those ones that are on the surfaces. Okay, then between the phospholipid molecules, cholesterol may be present. Now, cholesterol is a steroid. It is wedged between the phospholipid molecules in the plasma membrane, and it ensures there is stability. So it stabilizes the membrane fluidity. Okay, so this membrane is like a fluid. That's why you are referring to it as the fluid mosaic model. Why fluid? The membranes are not static. They are not static sheets of molecules locked rigidly in place. No, the membranes are held primarily by weak bonds. These weak bonds are called hydrophobic interactions. They are weaker than covalent bonds. Now, because of that weakness, the molecules, the component parts of the plasma membrane, have the ability to drift. They drift laterally in the plane of the membrane. It is a bit rare for these molecules to flip-flop, that move from one layer of the phospholipid to another. That's a bit rare, but it could happen. Now, the steroids, cholesterol, are supposed to stabilize. But remember, membranes must be fluid to work properly. If they solidify, their permeability is affected in one way or the other. Now, if permeability is affected, it means the cell can no longer get vital nutrients that it requires. Now, in very cold environments, membranes tend to solidify. So that means organisms that stay in cold places have adaptations to avoid their membranes solidifying. Once they solidify, permeability is affected. Okay, and that we shall discuss that one later. The presence of unsaturated hydrocarbons in the tails, where you have organisms living in very cold places, you have more unsaturated hydrocarbons, and that means that the tails do not fit together, and they will delay to solidify. Okay, looking closely at our drawing there, we looked at it in the beginning. Please, you should be able to identify the different component parts and take note of the hydrophilic and the hydrophobic portions of the phospholipid. Okay, the mosaic bit indicates that the proteins are not regularly positioned. They are inserted in between. The arrangement is not very regular, it is irregular, and we talk about the mosaic. Okay, that mentioned, let's move to the next slide and mention this, that the phospholipids and intrinsic proteins or the integral proteins are amphipathic. My friends, the key word that you should note is amphipathic. Remember, your exams have objectives in paper 1, and this particular word could come up, so you need to know it. Amphipathic, what does that mean? When you have a molecule that has both hydrophilic and hydrophobic parts on it, then that molecule qualifies to be amphipathic. Now, you must identify which components of the plasma membrane are probably amphipathic. We are saying phospholipids and intrinsic proteins. So that means that the extrinsic proteins are not amphipathic, but these ones, phospholipids, and those that span across the intrinsic proteins or the integral proteins, those are amphipathic. Please take note of that term. The extrinsic protein molecules are globular. Now that takes us back to the structures of proteins. We have the primary structure, we have the secondary structure, we have the tertiary structure, and then the quaternary. Now the tertiary structure actually is also referred to as the globular. So we are saying the extrinsic protein molecules are globular and they are bound to the lipid layer surface in a loose manner. Please take note of that. Okay, the intrinsic protein, those that span across, are rather tightly bound to the lipid layer. Tightly bound. So there's a difference in attachment there. Please take note of it. The proteins and lipid molecules are arranged in a mosaic fashion. That means there is no regular way. Mosaic, they are simply inserted. We said that the proteins are dispersed and immersed in the phospholipid bilayer. They're just immersed. You don't know exactly. Some are extrinsic, others are intrinsic, others penetrate slightly and they attach to carbohydrates. The arrangement is irregular. So the fashion is mosaic, and hence the name fluid mosaic. Expect questions on fluid mosaic. Why fluid mosaic? Please, that is for our revision. In summary, what we have been going through, what are the components of the plasma membrane? That table there in this slide shows us the different components, like the phospholipids and where are they located. The cholesterol, which is that steroid I was talking about, where is it located and what's its basic function? Then you have the integral proteins like integrins. Integral proteins, those ones that span across, where are they located? We need to take note of that. Then the peripheral proteins, those are the surfaces. Where exactly are they located? Remember, we said amphipathic. Some are amphipathic, others are not. So you need to know exactly which ones. Then you have carbohydrates that are attached to proteins, forming the glycoproteins. They also have a specific function, like we shall see later on. But I'm encouraging you to use your textbooks, read through. Our presentation here just starts you off such that you master and own the information that you study. Okay. Functions of membrane proteins. In our slides here, in our discussion here, we shall mention 5 functions. But from different textbooks, you could go to 6 functions or even 7. So I'm encouraging you to add on. I've given you 5, study them, research on them, but also find out if you can discover the other 2. And if you have not discovered them, please ask your question. Use this online platform when you come back to school. Let us do this together. Okay, so in the functions of the protein, membrane proteins, the first one, membrane proteins are involved in transport, and we refer to them as transport proteins. Look at the diagram there. Okay, look at the diagram, you have are they hexagonal molecules there just for representation, and we have now closely seen the plasma membrane. Now those materials must cross from one side to another, and the proteins are assisting them to move from one side to another. So in that case, in that way, the proteins in the membrane, the proteins that form part of the plasma membrane, are referred to as transport proteins. Not all of them are transport proteins, but some of them are performing that particular role. Going through the information written, an integral protein, that's the one that spans across, those ones that are amphipathic, okay? An integral protein in the membrane may provide a hydrophilic channel across the membrane that is selective for a particular solute. It is selective, so not everything, not all solutes will go through, but some that fit probably that conformation, that shape, will be picked up and taken across. That means that our membranes are selective. They are not just allowing everything to go in, but they will allow some, or some will pass more easily than others. So the permeability issue comes in there, and we are explaining that there are certain channels that are allowed. Now, transport across the membrane may occur by facilitated diffusion. We know diffusion is a movement of molecules from where they are in high concentration to where they are in low concentration, down a concentration gradient. That process does not involve energy. It is a passive process. Okay, so we are saying facilitated diffusion because the membrane protein is helping, is facilitating in the movement of particular ions, particular molecules across, either into the cell or out of the cell. So facilitated diffusion could also be referred to as facilitated transport or passive mediated transport. Remember, passive, it does not require energy. It simply requires a concentration gradient from high to low. Okay. So the process is spontaneous. We are saying a process of spontaneous passive transport of molecules or ions via specific transmembrane integral proteins. In that way, minus those proteins, these molecules won't be able to cross into the cell, and then the cell will cease to be the basic unit of life. You will not be able to provide the nutrients, the fuel, the material required for the cell to perform its function. Okay, so I hope the point of transport, the membrane proteins as transport proteins, is well understood. But not all materials cross into the cell or out of the cell by facilitated diffusion or simple diffusion. No, some require energy to be involved. So there must be another type of protein that will require energy to allow it pump ions across. Now, in the next slide, we say transport proteins known as pumps hydrolyze ATP as an energy source to actively pump substances across the membrane. So these substances will cross against a concentration gradient, probably from low concentration to high concentration. But you must use energy to pump them across. And those are special membrane proteins called pumps. In the intestinal mucosa, that is walls of the intestines, especially in the small intestines, and also in the proximal tubule of the nephron, are glucose transporter proteins. Such proteins in the kidney contribute to renal glucose reabsorption. My dear friends, if you find a trace of glucose in your urine, there is a problem. We talk about diabetes mellitus. I would like you, in your free time, to get a sample of your urine, put it in a test tube, add Benedict solution, warm, warm, warm. If you get color changes from blue to green and probably to orange through yellow, then it means reducing sugars like glucose are present in your urine. Once reducing sugars are present in your urine, there is a problem. There is a huge problem. You have issues with your pancreas that is not producing enough insulin, and that means you have some of the glucose ending up in your urine. But we have special transporter proteins around the cells in these tubules that are supposed to pick up this glucose and pump it back into the blood. Okay, 100% of the filtered glucose in the glomerulus has to be reabsorbed. Why are we talking about reabsorption? Because the first absorption was in the small intestines. Now this one is the second absorption, so you talk about reabsorption. So 100% of the filtered glucose in the glomerulus has to be reabsorbed along the nephron. Now, in this free time, you don't call it free because you're busy at home. Read through, look at urine formation, study the nephron, draw it, use your O-level information to start gaining information on this, because ultimately you must study excretion, and there you will see reabsorption. You're going to see reabsorption by active transport. Now, don't simply say active transport. That means you have special proteins called pumps that will pump this glucose back into blood, because you'd expect the concentration of glucose to be higher in the blood compared to the glomerular filtrate. Now, if you have a lower concentration in the filtrate and it's being taken into blood which has a higher concentration, energy must be involved, and these proteins come in, and that is one of the other functions of the plasma membranes. Okay, then when it comes to respiration, when you will be studying respiration, three parts of respiration: glycolysis, the Krebs cycle, and the electron transport system. Glycolysis, that one is seen is seen in the cytoplasm. It is basically in the cytoplasm, and there is breakdown of glucose, the hexose sugar. I think in our chemicals of life, you've been going through this. Our hexose sugar, which is 6 carbon, is broken down into a non-sugar component that is 3 carbon. And that non-sugar component is called pyruvate. Now, pyruvate is charged. It has a charge, and charged particles do not easily cross membranes. Now, how does this charged particle that is required to enter the mitochondrion such that it continues to release important raw materials that will end up releasing a lot of energy in the form of ATP can occur? So we have a special protein in the membrane of the mitochondrion. That protein built into the mitochondrial membrane translocates or moves. It picks up your pyruvate and takes it into the mitochondrion. Now you can see this membrane protein again involved in the transport of molecules or substances. Okay, it passes it into the mitochondrion or the mitochondrial matrix from the cytosol or cytoplasm, like I have mentioned. Second function is in enzyme activity. What is an enzyme? It is some molecule that is protein in nature. It is having a 3D structure conformation. In the primary structure, remember, a protein is made up of amino acids. These amino acids, if you're talking about a primary structure, are linearly arranged. They are far apart. Now, some of them have catalytic properties. Now, in that linear fashion, they are a bit far from each other. They cannot perform the role of catalysis. Catalysis. I don't know where that word exists, but it should. Catalysis. To catalyze. Okay? Now, if this primary structure is folded into a secondary structure, you could have two polypeptide chains lying side by side, or they are they are wound around each other to form a helical structure. Still, the catalytic amino acids will be far away from each other. But if we folded the secondary structure into a tertiary structure, then the catalytic amino acids are brought into close proximity. Now, those catalytic amino acids close together will form what is referred to as an active site on the enzyme. Now, that enzyme lowers the activation energy and makes reactions occur a little bit faster than they would if the enzyme was not present. Remember, enzymes are only produced in living cells. Now, looking at this drawing or this diagram here, or this picture in the slide, you have, I will refer to shapes. Okay, you have a rectangle that is green, then you have a triangle, and then you have a circle. Those are I could say substrates and products. Then in the plasma membrane we have two integral proteins that are spanning across. But look at the side that is at one of the sides that constitutes what you call the active sites. They have two different shapes. The active sites have two different shapes into which particular substrate should fit such that the reaction can occur. We are going to form a substrate. Yes. Okay. Now let's continue a bit. You have this rectangle that should fit. Look at the arrow. The rectangle should fit into the active site that is of a particular shape. Now, once that happens, the substrate is then broken down or transformed into a different shape, which is now a rectangle that should then fit into this other active site. You can see the shape. Now, this rectangle could not directly go and fit into this other enzyme. It has to first be transformed into a rectangle for it to be able to fit into this other active site for a round or circular product to be formed. Now, it is this circular product that probably is required. Now, in this case, we are having a series of reactions. Now, these proteins are built into the plasma membrane and they are held together. Remember, we said the integral proteins are tightly bound, so they are held in that position such that reactions that have sequential steps can occur easily. Now, let's go through the write-up. Some membrane proteins are enzymes, and I think we now know what an enzyme is. With their active sites exposed to substances in the adjacent solution, which is the cytoplasm. It could also be extracellular. Several enzymes may be ordered or positioned in the membrane as a team that carries out sequential steps of a metabolic pathway. Look at teacher S.5 and then teacher Baguma. Probably it is end of term and we are supposed to serve you with your reports. But because we are held up by time, we have not yet packed your reports properly. Now, Mr. S.5 is the class teacher. Me, I'm coming in also to help him out, such that after that we can go and hang out somewhere else. But we need to ensure that we serve you as quickly as we can. So what I do, I pack the reports, and then Mr. S.5 provides maybe he signs against and then hands to a specific student. So in that way, we shall be able to serve you all in the shortest possible time. We are like enzymes. But if I'm seated here and Mr. S.5 is seated down at the boys' end, probably near the gym, now you finish with me, then you have to walk across the playground to Mr. S.5 to get his signature. That will take time. However, if we are placed together side by side, then the reaction is going to occur very, very fast. I hope that explains what we are seeing here and one of the roles of the membrane proteins. Okay. For example, the proteins embedded in the inner mitochondrial membrane involved in the electron transport chain. As you are moving these electrons from one point to another, we have proteins in the inner membrane of the mitochondrion that are playing certain roles. Some are enzymes, others are carrier molecules. Okay, but because they are in close proximity, a reaction is able to occur to its completion fast enough. Okay, so enzyme activity is another role of the membrane proteins. The third one, cell to cell recognition. Man, if you look at how life happens, you just marvel and accept that really there is a being that is really supreme. You are saying a cell is the basic unit of life, but you also know that a group of cells forms a tissue and a group of tissues forms an organ. Now, what brings these cells together to form the tissue? You cannot bring a cell that is supposed to be in the nose with cilia close to a cell that is supposed to be on the skin. Maybe a cell that is supposed to be in the small intestine. The two cells are different. They cannot form a tissue. Tissues should have cells that are structurally similar. So how does one cell know that this one is the one that we can link with? So cell to cell recognition, that role is also performed by membrane proteins, especially those that have an attachment of a carbohydrate. Now it could be shaped in the form of a fork, and that should be able to fit into an adjacent cell. Now, once it fits, then those two cells can lie side by side, and that means you are going to begin forming a tissue. So we note that a cell's ability to distinguish one type of neighboring cell from another. This occurs when complementary molecules or on opposing cell surfaces meet. Some glycoproteins serve as identification tags that are specifically recognized by other cells. Cell-to-cell recognition is important, for example, when you are sorting cells or in the sorting of cells into tissues and organs in the animal embryo when we are still in our mother's wombs. During embryo formation, how do cells recognize? And now the plasma membranes, they play a significant role in that aspect. The fourth function of the plasma membranes, they are intercellular junction. Membrane proteins of adjacent cells may be hooked together in various kinds of junctions. Look at a junction. What's a junction? Where two roads meet. So that point of meeting becomes a junction. It could be a T junction or a Y junction. So using that English word, relate to what is happening. Look at the drawing. The two proteins lie side by side and then they form a junction. Okay, we are saying intercellular junctions are specialized regions of contact. But if you look at this drawing, one is spherical, the other one has a component slightly curves in to form a dome shape. Now this spherical part fits into the other one, and that means these cells are going to lie side by side. That point of contact is referred to as a junction, and it is again only made possible by certain specific membrane proteins. Okay, what is the use of these junctions? It's mainly adhere or contact to glue cells together, but the other one, interaction, communication between cells at that point where they have made contact. No cell is an island, especially if you're talking about multicellular organisms. There has to be some element of communication. Okay, so those contact points are referred to as intercellular junctions, and we have types of intercellular junctions. You could mention them, and in the next slide we give briefly the functions of each. Desmosomes, the tight junctions, the gap junctions, adherence junction. Please pronounce them in a way that allows you to spell them correctly. Desmosomes, tight junctions, gap junctions, adherence. But remember, they are for contact and interaction. In the next slide, we try to give the functions of each component, each type of intercellular junction. In your notes that I sent you earlier, these functions are spelled out. So we shall not dwell on them much because of time. But for example, the desmosomes link adjacent cells together and enable communication and transport between the two cells. The tight junctions prevent movement of integral proteins. They keep them in position. Okay? Because these integral proteins could be enzymes. You want them to be in that particular location. So you need a tight junction at that point. They could also be involved in maintaining polarity. Now, if these proteins were to change, remember we said amphipathic, you need to maintain particular polarities, a positive side or a negative, the non-polar and the polar. If they were moving up and down, they could interchange, and that would be problematic in one way or the other. Okay, so the gap junctions try to go through, and then the adherence, also look at them. You'll also probably mention them under histology, and also when you are dealing with transport, locomotion. In this slide, we try to present to you the location of these different junctions. So you study the drawing below and locate the different intercellular junctions. I think we have looked at the functions. These are about 3 cells, and you can see points of contact between these cells. You have a tight junction there, adherence, desmosomes, the gap junction. Please study them closely. You do not have to go into details for each of them, but at least you should appreciate that that is only made possible by the membrane proteins that we are discussing. In the next slide is the 5th function of the membrane proteins, signal transduction. When you convert a particular signal into something else, that could roughly mean transduction. Okay, you are having light energy changed into heat. You are seeing kinetic energy converted into something else. You could have materials, equipment for that, but we are saying signal transduction. The signal could come in the form of a hormone. Then the response, the effect is something far different from what the signal looked like. So signal transduction, we're again seeing the role of membrane proteins in that. Through the information that is written, cells have membrane proteins that are receptors. A receptor is an area that receives, okay, receptors, and they bind to signaling molecules. For example, hormones. A hormone like testosterone produced in the testicle has its effect, for example, on the chin when beards start growing, a secondary sex characteristic. Now look at the position of the chin and the position of the testicle. How are the two related? So testosterone, the hormone, is poured into blood. Then blood performs its role of transportation. But when this hormone, testosterone, reaches certain cells in the chin region, then you will have specific receptors on the cells in the chin region that into which probably the hormones will fit. And once they fit there, they trigger a response in the cell that results into the growth of a beard. Now, the other one was a hormone. Now, the effect is growth of beards, the development of the chest muscles, deepening of the voice. There is some element of transduction. Okay, I would like you to read more in that area. Signaling molecules are permitted to affect cell function without actually entering the cell. Okay, in this other slide, I give you one particular example. You have acetylcholine receptors, which are green. Look at the picture properly. These circular green things in the cell membrane there, close to the phospholipids, green. When you do not have acetylcholine, which is the signaling molecule, the pore here is closed. But once acetylcholine, which is blue, comes and attaches to the receptor that was green. Look at the green here in the middle one. But now in the position of green, you are having the blue acetylcholine. So once that signaling molecule attaches to the receptor, it causes the protein to open up, and the red important ions are able to move across into the cytoplasm. There is activation, pores open up, and what is required crosses. Nothing can cross until the hormone or this acetylcholine, this chemical, attaches to the receptor. Once it has attached, then there is transduction, pores open up, and then materials end up going into the cell, and then you're going to have a particular response. Okay, so read through that. That is a specific function of the membrane protein. So an acetylcholine receptor, which is green, forms a gated ion channel in the plasma membrane. This receptor is a membrane protein with an aqueous pore, meaning it allows soluble materials to travel across the plasma membrane when open. When no external signal is present, the pore is closed, like the picture, the picture in the center. When acetylcholine molecules, which are blue, bind to the receptor, this triggers a conformational change. There's a change in the shape. So that conformational change opens the aqueous pore and allows ions, which are red, to flow into the cell. That is an example of signal transduction. Members, I thank you so much for listening. I hope you received your notes. Please write them in your books and continue researching and researching and researching. Remember, the wider we read, the more area we cover, the lesser the pressure, because there's a relationship between pressure and area. Pressure is inversely proportional to the area. If you can cover much, that means you cover a wider area, the pressure on you becomes low or less. And in that way, you are able to internalize your concept and be as confident as possible, and you should be able to perform well. Issues at hand, there is coronavirus disease. This disease is caused by the coronavirus, the COVID-19. The beauty about it is that we are the vectors for this virus. So make sure you don't transmit the virus. Wear a mask. Purchase yourself a mask whenever you go out. Wash your hands. Sanitize regularly. Cover your mouth. Don't sneeze in the open. Use your elbow and things like that. Please stay safe. We need you safe. You have numbers there in case you're not feeling fine. Call those toll-free numbers and get urgent treatment. We thank God that we are doing a little bit better than other countries in the fight against COVID. Please stay safe. We want you alive. Thank you very much. Your presenter, Baguma Gerard Mutiba.